And so what we've got are orthogonal chemistries. They can coexist in the test tube, and they can coexist inside the embryo. So as a result, if we want to target these, we can use probes that have this initiator sequence. The part that's in the sort of purple color in here is the complementary sequence, so it allows it to see one of the RNAs. And then when we add in the hairpins, we get on a leash a whole bunch of dyes assembled. And so what this allows us to do is to add all the probes, wash them away, add all the hairpins, and you can do up to 8 genes at a time the way it is right now. And if you do that, you end up with something that's a little bit invisible in today's light. But what you should see as this movie plays through is in C2 whole mount embryo in which 5 different genes are stained. And if you had eagle eyes, you might notice that the signal is a little bit granular. That's because the dyes stay on a leash. So each one of those puncta is one messenger RNA. So for rare transcripts, things that aren't expressed very high, the hope is that we can go in and actually count how many messages are within the cell. So go over to a sort of digital version of in situ hybridization and understand what co-varies within individual cells. The resolution is very nice. You can see an individual endothelial cell as it's squeezing between 2 muscle cells, and you can see the nascent transcript in the nucleus that's being run off of the DNA, and you can see this thin lamellipodium that's only a micron or 2 thick is carrying the message with it in between, and you're not at all confused between the red that's in the endothelial cell, going to make a blood vessel, and the muscle cells to the side. So we got cell autonomy, we've got the multiplexing, and it's turning out to work okay on everything from human biopsies to sea urchins and beyond. So what we're hoping is this is a technique that could be a general utility to allow us to ask these multiplex questions, multiple questions about the same biopsy, about the same regenerating cell, the same stem cell that's being reprogrammed, or whatever we want to go after. There's a website that has the recipes and things like that. And we've been improving the technology and offering probes that are already troubleshot. And the cost is mentioned here because the website will even help you order probes from IDT, one of the major supplying houses of synthesized oligonucleotides. So this approach is allowing us to really try to exploit the sort of unexpected lineages we're seeing, the unexpected contributions. And of course, those are the things we want to be able to exploit if we want to think about regenerative medicine. We don't want to just do it the way the textbook says. If it works just the way the textbook says, we should give up all hope because the textbook doesn't say you can regenerate a new airway or whatever it is that we need. So we want to be able to understand the lineages and the interactions. We've got a number of different techniques that are letting us carry the grain to single cell analysis. And as we're doing this now, we're realizing that what we really need to do is to carry this to a molecular question within the cell. And I want to just show you a couple things that have been going on in the lab that try to carry the cellular question down to the molecular question inside of it. So let me just go forward. One of these technologies is called fluorescence correlation spectroscopy. It's a technique that biophysicists and chemists have used for a long time to understand how quickly a molecule can move. And the way that it works is instead of taking a laser scanning microscope and scanning the laser beam around, you just park it in one place. And you park it and you watch. And as labeled molecules move through that parked focus, there's a signal, and then as they migrate out, the signal goes away. So what you see is this noisy trace as you park in one place that reveals when molecules come into the trap and when they go out of the trap. Now, how quickly they move will dictate how quickly the fluctuation is. How many there are in the cell tells you where this sort of autocorrelation curve intersects one of the axes. So by just parking it there, you could count how many of a given molecule is around. So if we were in the coffee session and I counted who was underneath this lamp and we were all milling around, just by watching the different heads going under it, I could do a pretty good count of how many people were in the room. good to a couple percent. And that's good enough for the sort of thing we're trying to do. I could also find out how quickly we're milling around, because if one person stayed under it for a long time, then these wiggles would be slower. And on top of that, if David and I had different color hats on, and we were holding hands and always moving together, then we'd be able to tell the fraction of the molecules that are bound to one another, what their binding constant is, and count the bound versus free, and get some molecular questions. This has now been a nice collaboration between Andres Collazo and Lei Trin in the lab to try to go into some gene traps. We've got, in this case, it's a G protein. The G protein is much more mobile in cells where the second messenger mechanism that it's involved in is active. And what you can see, if we just go in and ask with the gene trap, where is the gene protein, it's beautifully expressed only in the trigeminal neuron and in the developing hair cells of the inner ear. But if we look in with FCS and ask, where is it that that molecule is actually doing something? Where is it that they're signaling? What you can see is that in the red bar, the hair cells, they're dramatically more mobile. So that's where this G protein is actually doing some signaling. In the trigeminal neurons, there's, if anything, more of the molecule there, if we count how many molecules are there per cell, but they aren't involved in a molecular dialogue. And so this is allowing us to go from just saying where a molecule is to where it's actually doing something. And we're now hoping to use this in questions like stem cell reprogramming to ask not just what's expressed there, but what's doing the deed of reprogramming the cell or of cueing it in. Lei's now using it to look at questions of chromatin structure, and so we can get into the epigenomics and understand the mobility and the action of factors that are involved in opening up chromatin and of splicing the RNA together to make the mature message. Now, a related question is to go into cells that look remarkably similar. And this is some work that David and others at Children's Hospital played a big role in, from Laki Pantazis and Nico Plashta when they were in the lab. We'd like to be able to go into something like a developing embryo and predict ahead of time which cell thinks it's the stem cell. They look identical. If you try to figure out which one is which by staining for different expression levels of key factors like OCT4, what you see is that there's brighter cells and there's dimmer cells, but the brighter cells are just as likely to not become the stem cell as the dimmer cells are. So clearly expression level isn't good enough in predicting which cells will go to the inside and eventually make the inner cell mass that'll go on and make the embryo. Okay, so what Laki and Nico decided to do is develop a fusion of OCT4 and other key transcription factors with a photoactivatable GFP. And with our 2-photon microscope, we can go into individual nuclei and uncage that transcription factor. And that allows us then to follow the mobility of the transcription factor in this nucleus versus any other. And when they did that, they were able to see that different cells, even though they had varying expression levels of OCT4, as much as 5 or 10-fold different levels, some of the cells retained the OCT4